The next stain that we're doing is the Endospore stain. So we did the gram stain for looking for gram-positive, gram-negative. Within those gram-positives, you can have the acid-fast cells, like the mycobacterium that we talked about last time. Now we're going to look at endospore staining. The endospores are produced by mainly gram-positive rod-shaped bacteria. The reason that they're mainly produced by bacillus bacteria is because they are generally large bacteria. Small, tiny bacteria do not have room to produce endospores within them. Now the endospores are resting, they use the word cell, but they're not technically cells because they're not like actively dividing and growing, okay? They are a resting state, kind of like the, not so much as being in hibernation, but you can think of it as that time where they're not actively doing anything, okay? One of the groups of bacteria, the genus that produce endospores, are usually going to fall into the Bacillus genus or the Clostridium genus. Ones that are pathogenic, we find in that Clostridium genus. If you've ever heard of botulism, tetanus, gangrene, those types of things, those diseases are caused by bacteria in that genus Clostridium. So Clostridium botulinum can cause botulism. Tetanus, things like that, are in this group. So it's really important to know if a bacteria produces an endospore. Now that bacteria that produces an endospore can produce toxins later that make you sick, like with botulism or tetanus. The bacteria itself is going to be a large bacteria, and the endospore is going to form within the bacteria. The endospore is very, very, very resistant to environmental factors. It is basically made up of DNA, so the DNA of the cell, and then it is wrapped in layers of membrane and wall, and it's extremely thick. If you look at it in a picture of a book, you'll see that the wall of the endospore is twice as thick as the wall of the bacteria. Now, I mentioned that they're resistant to environmental factors. So when a bacteria, such as a bacillus or a clostridium, is about to die or getting close to its period of extinction, of expiration, I guess you would use, it's going to start producing this endospore. So the lifespan of a bacteria is going to be like 48, 72 hours. It doesn't live much longer than that. It will either reproduce before that time. Also, if it produces endospores, it will do that. So when a bacteria is, heading the end of its life, and it's ready, getting close to dying, it's going to produce that endospore so that it can pass its DNA on to another generation even though it's gone. Okay? So this endospore will form, and then as it gets older and older and older, the endospores forms inside it, and then the cell will burst when it dies, and the endospore will be released to the environment. Now, it's not like a spore in a fungus where you see it floating in the air. It will remain in the soil, it can remain in a can or something like that for years and years and years. There could be an endospore sitting in the soil for 30 years, and then later it'll be activated. So when the environment changes and conditions become favorable, that endospore will actually start germinating into an actual cell. Okay? So say you have a bacteria or a colony of bacteria that have released 100,000 endospores, and they're sitting in the soil, and it's been really dry. They're just going to sit there. But then a nice rain comes, and the rain will activate those endospores, and they will germinate. And if you've ever heard of tetanus, you step on a rusty nail and you can get tetanus. Well, what happens is that nail is rusty because it was wet. Well, during that time that it was wet, those Clostridium tetani endospores have been reproducing and producing this toxin. And when you step on that rusty nail, that toxin goes up to you. If you haven't had a tetanus shot or an inoculation for tetanus, that can go and block your relaxation pathways. So that it's really important to understand if bacteria produce endospores. So with a bacillus, you would do a gram stain, or after you've done a gram stain, you realize you have a bacillus bacteria, then you would do an endospore stain to see if it's an endospore-forming bacteria. Then you can start going further in the classification. The endospore stain is similar to the acid-fast stain in the fact that we're going to use heat. The difference is there's only the the first stain and the second stain. The primary stain in this case is not staining the outside wall of the bacteria, it's actually staining the endospores. Then your counter stain will stain the wall of the bacteria. There's no decolorizing step, okay? So just as we did with the acid-fast stain, we're going to put our slides that already have a fixed culture on them that are labeled, mine aren't, that are labeled on our heat. And once again, we don't need it rapidly boiling, we just need to see steam coming off of it. So I can see the steam, the condensation forming. As we did with the acid-fast stain, we're going to drop the stain on there. Oh, the first stain in the Endospore stain is called malachite green. So we're going to put our malachite green on there, making note not to get too much in the water. That is going to steam for the 5 minutes. During that time, you can do whatever else you need to do. Remember to watch it. So don't just walk away and come back 5 minutes later. You want to watch it. If it starts to dry out, you add more stain. Once again, I don't want it running over and have a bunch of green water, okay? So that's going to steam for 5 minutes. The steam allows the stain to penetrate the endospore wall. Once it penetrates the wall, it can't come out. So then when you rinse it, it remains in the wall of just the endospore. The actual cell itself will lose the stain and be colorless, okay? So after our 5 minutes, we're going to gently take them off. Once again, you'll note I can just grab them with my fingers if you have the right size beaker, and the water's not that hot, okay? Turn that off because I don't need it anymore. We're going to rinse with the water as we've done before. Now, if your slides get extremely hot, that probably means that your water was too high, but if they do, don't rinse them right away because they're going to crack, okay? Mine weren't too hot. If they're cool to the touch, you're good to go. So rinse them really good like we've got. Once again, don't be afraid to rinse, rinse, rinse. You can't really over rinse. We have no decolorizing step. We're going to go immediately to the counter stain, which is safranin. You saw safranin as the counter stain in the gram stain, okay? So what we're looking at here is not necessarily two different colored cells. If you have a cell that is endospore positive, meaning it is an endospore producing cell, you will have the endospores themselves green and the cell membrane or the cell wall will be pink. So they're kind of Christmas cells, they're red and green. Now you may also see, so after that's set its time, I'm going to rinse. So again, rinse, rinse, rinse. Don't be afraid, you can't really over rinse. Now you don't want to spray directly on the culture. And as we did before, we're going to blot dry. A lot of these procedures that you've learned, we're repeating, okay? Smear, fix, then stain. After we stain, we blot dry. Now this is going to be different on the next two stains that we do. But as of yet, everything that we've done pretty much follows the previous procedure, okay? Always make your smear and fix it before you start, okay? So we blotted those dry. So I was saying that at the end of the stain, you're going to have a green endospore and a red cell. Now the endospores may actually be within the cell, or they may have been released. So depending on the age of the culture, if it's early in the life cycle of the culture, they may be inside. If it's late in the life cycle of the bacteria, then it may actually be released. So be prepared to see them either within the cell or outside of the cell.